Good morning, everyone. I'm delighted to be a part of this very prestigious meeting, and I would like to thank all the organizers and also my dear friend Tarek for their kind invitation. So what I will try to do in the next 30 minutes is to talk about the role of PGTA in an evidence-based IVF practice. So this is a typical paradigm for an IVF treatment. So we stimulate the ovaries, we collect the eggs, we fertilize them, and we transfer the resulting embryos. The patient either gets pregnant or does not get pregnant. Some of the patients who do get pregnant will deliver their babies. Some of the pregnancies will end in a miscarriage. And after this miscarriage, some of these patients will drop out. Some of these patients will come back. And also the non-pregnant patients will also, some of them will drop out and some of them will come back for another cycle or transfer of their frozen embryos. So the basic outcomes that we are interested are pregnancy, whether the pregnancy continues and culminates in the delivery of a healthy child, and the presence of frozen embryos if we are going to transfer them at a future date. So we will be looking at cumulative delivery rates. Furthermore, we are also interested in the time that it takes for this patient to deliver a healthy child, which we call time to delivery, and also the costs that are incurred. So this is a typical IVF cycle in a typical IVF center. And I think everybody would agree that the success rate of assisted reproduction depends on the selection and transfer of the best available embryo into an adequately prepared and anatomically normal uterine cavity by an experienced provider. So these are the three basic necessities for an IVF cycle to be successful. Preimplantation genetic testing is invasive diagnostics with biopsy of polar bodies of oocytes, blastomeres of 8-cell embryos, or trophoblast cells of blastocysts. This is followed by indication-dependent relevant genetic analysis. There are also some coming new developing techniques such as semi-invasive pre-implantation genetic testing with aspiration and DNA analysis of the blastocele fluid, and non-invasive techniques such as analysis of spent culture media. But basically today we will be talking about pre-implantation genetic testing for aneuploidy as it is performed. So now we are in the version 2 of this pre-implantation genetic testing. Version 1 actually had a severe plane crash, and after that, it was totally abandoned. And now we are experiencing some of the problems that we have experienced with version 1, and we are now experiencing them with version 2, and I will touch upon these in my talk. So today, the way it is practiced is the cells that are removed from the embryo are trophoectoderm cells, and advanced testing platforms are used to do the genetic analysis. So these advanced testing platforms may be SNP arrays, CGH, RT-PCR, NGS, low-density NGS, and whole chromosome amplification followed by NGS. So these platforms by themselves have different advantages and disadvantages, and they may test for some of the problems in the embryo, but not others. And today, most commonly, the preferred platform is next generation sequencing. In its simplest form, PGTA is a selection method employed through diagnostic testing of an embryo. So it should be subject to all of the relevant problems, shortcomings, advantages of diagnostic tests. And especially in preimplantation genetic screening or preimplantation genetic testing for aneuploidy, the prevalence of the condition that we are testing heavily influences whether we get true positive, false negative, or true negative rates. So the prevalence of the problem that we are investigating is of extreme importance. And we know that most of the randomized studies up to date have been performed in patients with a good prognosis, meaning that they have many oocytes and many embryos available, which do not reflect the true population that we are interested in. So why do we do PGTA? We aim to increase the pregnancy rate. We try to reduce miscarriage rates. We try to reduce multiple pregnancy rates. We want to reduce the rate of malformations due to chromosomal abnormalities, and we want to decrease the time to pregnancy with reduction of multiple ART treatments. It's obvious that if we can select the embryo that has the highest potential to implant, then the time to pregnancy will decrease and the patient will reach the happy ending much before than others. And which population will most likely benefit from PGTA? Just by judging from the previous slide, women in their reproductive years, in their late reproductive years, meaning these women will have more aneuploid embryos, women with repeated implantation failures, women with repeated miscarriages, men with severe male factor infertility, women undergoing single embryo transfer, which I will talk about later because most of the embryo transfers now performed throughout the world do not include many embryos. For example, in Turkey, we are allowed to transfer only one embryo in patients who are less than 35 years of age. And this is true for most of the European countries. And now it is slowly, although steadily, becoming the norm also in the US. Women undergoing PGT-M for monogenic diseases. So you may combine PGT-A with PGT-M after selecting the embryo that doesn't carry the monogenic genetic abnormality. You can also test with PGT-A to see whether they are euploid or aneuploid. And some have suggested that PGT-A should be applied to all women across the board undergoing IVF treatment. The assumptions are the elimination of aneuploid embryos prior to transfer will improve the outcome of IVF, which appears to make sense. A single trophoectoderm biopsy at the blastocyst stage is representative of the whole trophoectoderm. The inner cell mass. And trophoectoderm ploidy reliably represents the inner cell mass, and ploidy does not change or self-correct downstream from blastocyst stage onwards. When you look at the biology of PGTA, if you have a euploid embryo, you will have a euploid blastocyst, a euploid inner cell mass, and a euploid trophoectoderm. If a meiotic error happens in the embryo, it will involve the inner cell mass and the trophoectoderm, and you will have a uniformly aneuploid blastocyst. If there are mitotic errors leading to genetic problems, then you will have a mosaic embryo with mosaic cells in the trophoectoderm, in the inner cell mass, or one or the other. So one assumption of PGTA is that a single trophoectoderm biopsy at the blastocyst stage is representative of the whole trophoectoderm, and trophoectoderm also represents the inner cell mass. So let's look at different scenarios. For example, this is a uniformly euploid embryo. So you remove these cells over here, you send it for genetic testing, and the result will then come back as a euploid embryo. and trophoectoderm biopsy will have correctly reported euploid embryo. And the same is true for a uniformly aneuploid embryo. So if you remove these cells, the biopsy result will come back as aneuploid, and aneuploidy is uniformly within the embryo. So it correctly reports an aneuploid embryo. However, if you have a mosaic embryo, a euploid ICM with a mosaic trophoectoderm, so you see the ICM over here in green, which is normal, euploid, but the trophoectoderm contains euploid and aneuploid cells. And if you remove these aneuploid cells over here, then you will be incorrectly labeling this embryo as aneuploid, and you will be discarding this embryo, and you will not select it for embryo transfer. However, if you would have, it should result in a normal pregnancy. You may also remove the euploid cells over here and from the trophoectoderm, euploid and euploid inner cell mass, and together this will be correctly predicted in this case. It just depends on where you take the biopsy. And unfortunately, when you look at the embryo, you are not going to be able to judge just by morphology alone where the euploid cells and the aneuploid cells actually lie within. So this is an embryo with mosaic trophoectoderm, but an aneuploid inner cell mass. So if you remove these normal cells, then you will be reporting incorrectly that this embryo is normal. However, this embryo is not normal, and when transferred, it will either not result in a pregnancy or it will result in a miscarriage. Therefore, you are destined to have false negative and false positive results. This is an aneuploid ICM. If you remove the euploid cells, you will false negatively report that the embryo is normal. However, this embryo is normal with a mosaic trophoectoderm, and if you remove these cells over here, then you will report that the embryo is false positively abnormal. So you will be discarding this embryo, whereas you should have transferred it, and you will be transferring this embryo, whereas it should have been discarded. And then there is the problem of mosaic embryos. So mosaic embryo, mosaicism, is considered to be a normal feature of human embryos, and it appears that the rates do not increase with female age. And mosaic embryos are known to self-correct through clonal depletion of abnormal cells and eventually produce normal offspring, and transfer of these embryos can result in normal pregnancies. So this is a very recent study, and it was done on 1,000 1,000 transfer of mosaic embryos, so in 1,000 patients. And they mosaic embryos, they graded them as high-level complex mosaic embryos all the way up to segmental mosaics. Low-level 1 chromosome, low-level 2 chromosome, low-level complex, high-level 1 chromosome, and high-level complex mosaics. And you can see over here, this is the implantation and this is the ongoing pregnancy rate. You can see that the segmental mosaic embryos and embryos with low-level mosaicism approximately result in the same implantation or very similar implantation and ongoing pregnancy rates, as opposed to embryos having complex mosaic structures will result in far less implantation and ongoing pregnancy rates. Still, however, even the embryo is a high-level complex mosaic embryo, there's approximately a 15% chance of a normal ongoing pregnancy. So the other assumption that we make when we offer PGTA to our patients is that the trophoectoderm ploidy reliably represents the inner cell mass. and ploidy does not change or self-correct downstream from the blastocyst stage onwards. So this is actually what happens. So you have a blastocyst that will implant, and the trophoectoderm will eventually become the placenta, and the inner cell mass will eventually become the fetus. So the questions are: does a single 6-cell trophoectoderm biopsy reflect the whole trophoectoderm? And does the trophoectoderm chromosomally reflect the ICM? And how much does ICM self-correct downstream from blastocyst stage onwards? So we know that the embryos actually rescue themselves. So this is a meiotic non-disjunction resulting in a trisomic embryo, with some of the blastomeres being trisomic and some of them are disomic. And eventually the embryo can allocate its normal cells to the inner cell mass and abnormal cells to the trophoectoderm, resulting in the placenta. And this will result in a normal pregnancy and a healthy baby. So I will show you 2 studies coming from the UK, from the group of Magdalena Götz, and both are mice studies. And granted that mice are not humans and mouse embryos are extremely different than human embryos, still these studies give us some idea whether this aneuploidy can be self-corrected, with eventually having a normal developmental potential for that index embryo. So what this group showed was that when they tagged these embryos fluorescently, these embryos are embryos having both aneuploid and euploid cells, and these are induced aneuploidy in the mouse, because in mice do not produce aneuploid embryos, so it's extremely rare. These studies have been criticized because they have been criticized due to the fact that mice embryos are completely different than human embryos, and the results obtained cannot be extrapolated to humans. However, still, we know that in these embryos, abnormal cells or aneuploid cells undergo a much higher rate of apoptosis as opposed to cells that are normal, meaning that the embryo preferentially actually eliminates these cells and tries to rescue itself, resulting in healthy, normal offspring. And this study came out just last year from the same group. And what they showed was this correction process actually was an autophagy mediated apoptosis in this mouse model of chromosome mosaicism. So they used reversin and DMSO. So these are, and they made chimeric embryos, diploid and aneuploid. And then these chimeras, the diploid aneuploid chimeras, they removed the inner cell mass by immunosurgery and put that inner cell mass in a matrigel. And then they looked at that, they looked at the growth of the embryo, and they were able to show that these embryos, which were abnormal, actually preferentially eliminated their abnormal blastomeres. These embryos are chimeric, so they have normal and abnormal blastomeres. So they eliminated the abnormal cells much more compared to their normal cells. So these apoptotic cells were actually secreted into the surrounding media. And then when they transferred these cells into pseudo-pregnant mice and kept them there for 12 days, and then again removed them afterwards, they looked at whether these cells were also eliminated, the aneuploid cells. And even from stages from blastocyst onwards, after implantation, the embryo appears to be still able to correct itself, resulting in normal offspring. So what happens in humans? This was a study published in Human Reproduction in 2019. And what they did was they removed the trophoectoderm cells from the embryo, and then they looked at the concordance rate between the trophoectoderm and the inner cell mass according to the extent of chromosomal abnormality. So in embryos with single chromosome aneuploids, multiple chromosome aneuploids, or segmental aneuploids, especially with multiple chromosome aneuploids, most of the embryos were either aneuploid imperfect or perfect concordant. However, with single chromosome aneuploids, there was still an approximately 5% of these embryos were still not concordant, so they were normal. The inner cell mass was normal, and there was an imperfect concordance rate of 8%. When you look at segmental aneuploids, you can see that approximately 60% of these embryos have an discordant trophoectoderm and an inner cell mass. The study by Popovich that was published in Human Reproduction in 2019 actually tried to answer the question that was posed in mouse embryos by Magdalena Götz's group. So what they did was they cultured the embryos that were either donated, the excess embryos that had undergone PGTA and were either that were euploid, and embryos that were tested for monogenic diseases that were aneuploid. So and they put these embryos into the culture, and they waited for 7 to 12 days, and almost 90% of the embryos were viable if they were euploid to start with, as opposed to none being viable if they contained multiple chromosomal anomalies. You can also see that trisomic embryos fared the same as normal embryos, suggesting that these embryos are compatible with a normal implantation and a normal pregnancy. So the question is, can a non-selection study give us definitive answers, meaning that when we transfer the embryos without knowing whether they were aneuploid or euploid? Well, this study was published in 2020 by Emre Seli and Richard Scott's group, with Ashley Teagues being the first author. And what they did was they enrolled 402 patients, and these patients had single frozen blastocyst transfers, and all of these blastocysts had been subjected to PGTA, but the clinician providing the transfer was unaware of the result, so they did not know the result during the transfer. And after the clinical outcome was obtained, meaning that the babies were born, unblinding of PGTA results were performed, and they looked at the outcome of transfer of euploid embryos and aneuploid embryos. And what this study showed was, whereas 82% of euploid embryos implanted, half implanted when they were aneuploid, and sustained implantation and delivery rates were 0 in the aneuploid group, as opposed to 65% in the euploid group. So none of the embryos in this study that were aneuploid actually implanted. And the authors concluded that aneuploid single embryos do not result in sustained implantation and deliveries. Euploid single embryo transfers result in high sustained implantation and deliveries, and embryo biopsy at the blastocyst stage does not affect clinical outcome. However, this study has also been criticized because the study has been performed in good prognosis patients, women less than 35 years of age. And the prevalence of aneuploidy is dependent on age. And as I showed you in one of my first slides, that when you consider PGTA as a diagnostic test you also have to take into account the prevalence of the problem within the population. And variance in prevalence would affect the predictive values and limit the generalizability of the study to clinical practice in general. So it cannot be extrapolated to your typical IVF patients. This study also showed that when they compared this sustained implantation rates in patients who were biopsied and in patients who were not biopsied, this is a control group undergoing non-PGTA cycles during the same period of time. They said that both of these sustained implantation rates in both groups were the same. So they concluded that the biopsy procedure itself doesn't appear to harm the embryo. We actually have one study by Scott group in 2013 showing that there's an obvious reduction in sustained implantation rates in embryos that were subjected to blastomere biopsy as opposed to trophoectoderm biopsy. That doesn't seem to affect the survivability of the embryo. Well, are we sure that this is the case? Most of the patients in the Scott study were less than 35 years of age, and almost all of them had a good ovarian reserve, judging from the fact that they had many eggs retrieved, and all embryos were of good quality. So can the result of this study be extrapolated to women greater than 40 years of age? How about lower rate of blastocysts, and what happens after cryopreservation? These are the things that we do not know. Lastly, we will take a look at the RCTs in the literature. So I'm just going to talk about the RCTs with version 2. And as you can see, the ones in yellow are the RCTs that have been considered in previous meta-analysis and systematic reviews, but they actually contain many problems. For example, the RCT by Urman actually compares ESET with DET in the control group, elective set in the PGT group. The study by Yang just looked at the pregnancy rates after transfer of embryos in the first cycle. So pregnancy rates were judged by the denominator was embryo transfers, and they did not look at cumulative pregnancy rates. Neither did any of these studies over here. The study by Scott also suffered from the same problem. They looked at pregnancy rates, not intention to treat, but only per embryo transfer. Rubio study, biopsy was performed on blastomeres, which do not appear to be the case anymore. So we do not prefer blastomere biopsies. Esteem study, you can see over here, the biopsy was performed on polar bodies, and the preferred technology was only used in the STAR study, which I will touch upon briefly. So in this study, which appears to be a study that was done on very good prognosis patients, as you can see over here, over 50% were less than 35 years of age, and only 2% of these patients in the PGTA group had a low ovarian reserve. Just to give you an idea, the patients that we perform IVF cycles on, currently approximately 1/3 of them will have a diminished ovarian reserve in our center. And you can see that the number of mature oocytes collected, number of oocytes collected 18 and mature oocytes 14, and these patients had 7.5 blastocysts per retrieval. And just to give an idea, what happens within our clinic is we have approximately 1.5 to 2 blastocysts per patient overall. So this is our overall IVF population. And when they looked at the ongoing pregnancy rates per transfer, it appeared that PGTA fared better. But when they did an intention to treat patient population assessment, actually control group patients had a higher ongoing pregnancy rate for transfer. Only in patients between 35 years and 40 years of age, it appeared that PGTA within the intention to treat population resulted in a slightly higher ongoing pregnancy rate. But there are also several problems with this study. If you look at the consort, and if you include these patients over here, 84 who did not have euploid embryos, 42 with no euploid embryo, did not have 2 embryos, no euploid embryos, 6 with thaw failures, and also the same in the other group. And if you do another corrected analysis of the STAR trial that was done by Orvieto and Gleicher and published in Journal of Assisted Reproduction and Genetics in 2020, you will see that there is no difference between PGTA and the control groups in patients overall and in patients between 35 to 40 years of age. This study, this is the European study that was done, unfortunately, on patients removing polar bodies, and this is the study on advanced maternal age. And if you look at the women with live birth rates, you can see that there was no difference in between patients who were subjected to PGTA and patients who were not. So a Cochrane review was also done. We can safely conclude that the version 1 of PGTA with cleavage stage biopsy and FISH analysis actually did not benefit but harmed the embryo and decreased the live birth rates. And also blastocyst stage biopsy, the study published by Jensen, also was shown to result in a lower pregnancy rate in the PGTA group. So when you combine all of these studies, it favors no PGTA in an IVF cycle. So the studies that I mentioned before by Foreman, by Yang, by Scott were all excluded in this meta-analysis due to limitations, which I will touch upon in my next slide. But if you take the 2 studies done by polar body biopsy, the European study and the study by Mune with a blastocyst stage, both of these studies did not appear to show any difference in between PGTA and non-PGTA. Although the polar body biopsy study, the study by Verpost, showed a slightly lower miscarriage rate, the study by Mune did not show this either. So as I said, the 4 studies were excluded as the use of a single embryo transfer in PGTA group versus double embryo transfer in the control group. These 2 studies by Foreman and these studies by Scott, because they had reported on women that had an embryo transfer with no intention to treat. So there are limitations of the randomized studies. In IVF with PGTA compared to IVF without PGTA, a relatively large group of women will not have an embryo transfer, as all embryos are considered aneuploid or unsuitable. And if a study only includes women with an embryo transfer or only reports outcomes per embryo transfer, then the study will favor PGTA by design. And these studies, unfortunately, do not permit the drawing of any conclusions on the effect of the effectiveness of PGTA. And this has been addressed by several authors, as you can see over here. These authors heavily criticized also version 2 of PGTA and concluded that it is only an expensive add-on, which without any demonstrated benefit for the patient. Furthermore, there are financial aspects. For example, if you do PGTA in a young patient, the cost in US would be about $30,000 for having 1 euploid embryo, as opposed to if you take older patients, then the cost of PGTA will steeply rise, and just to have 1 euploid embryo available for transfer. And this study that was done by Somigliana's group in Italy showed that only in patients over 38 years of age, more than 3 or 4 blastocysts, then PGTA may become cost-effective, but not otherwise. So to conclude, we have to say that RCTs have been performed in highly selected good prognosis patients, and there is a more recent emphasis for measuring IVF success rates, which has shifted to cumulative pregnancy and live birth rates per cycle started or intention to treat. Embryos identified as abnormal by PGTA have been transferred, resulting in live births. Chromosomally mosaic embryos can be transferred, resulting in apparently healthy live births, and safety of trophoectoderm biopsy and possible harm and outcome of subsequent pregnancies are still being questioned. So in my opinion, PGTA is overutilized without any clear evidence that it is indeed useful, and more importantly, not harmful to the embryo and the patient. And more appropriately designed randomized trials are urgently needed, as always. And until then, caution is advised in recommending PGTA as a useful add-on in IVF. I would like to thank you very much, and also, again, thank the organizers for giving me this opportunity to participate in this very prestigious event. Thank you.